Hey y'all, I'm Sunny Shell, and tonight we're coming on with a new night and with some new improvements. But first, tonight's topic is biochemistry and abiogenesis. So sit back, buckle up, and let's get to the sunny spot on Skeptic Haven. Hey everyone. So, as you can tell, we're on a new night. We moved from Mondays to Thursdays. It seemed to fit a little bit better. But we also made one big improvement that I'm super excited to announce. We have a co host. And our co host is someone that I absolutely adore. I wouldn't want to co host with anyone else. So, let's introduce the new co host. It's Bumblebee.
Hey, Sunny. Yes, we got Sunny and Bumblebee now teaming up, and we're gonna be the Sunny spot. And I'm so excited to be part of this. And this isn't just like a one-off thing. This is you'll see me here all the time now. I freed up my Thursdays to come hang out with Sunny and hopefully talk as much science and philosophy and stuff as I can, and engage with as many new people. So thank you for inviting me to be part of your show, Sunny.
I am so thrilled that you said yes. And you'll see Bree on when I'm not on. For instance, next week I am going to be out of town at a conference, and Bree's going to take over for that so she'll be the host. So I'm really excited. You are fantastic. You know, I absolutely adore you. So thank you. So tonight we have our special guest. She is amazing. So let's go ahead and bring her on. It's Sciencey Melly. Hey,
Hi.
we are so excited to have you on.
I'm so excited to be on. You have no idea. I was posting all about this.
Yay! So tell us a little bit about yourself and maybe why you're in this field of science.
um okay, yeah, sure, sounds great. I basically I originally didn't want to be a scientist, by the way, at all. Like sophomore year of high school I was like hell no. And then I got into a geoscience class that like skyrocketed everything, and I was like, yeah, I'm gonna be a geology major and everything. It has changed so much since then. I entered college as an environmental science major because that was originally what I wanted to do. It was a whirlwind. I got basically rejected from environmental science, and then like for some reason the University of Texas at Austin was like, you know what we're gonna let you do though, we're gonna like let you have a biochemistry degree instead. And so I chose biochemistry. I got into that, and then I realized like halfway through I was like, I really don't want to work in like a lonely lab the entire time because I'm very social and I like love talking to people. And I was like, and also I don't want to do pre-med by any means. I'd cry. So I was like, forensics, we're gonna do that. And now every single time someone asks me why I'm a biochem major that's not pre-med, I just say because dead people don't talk back. So it's it's pretty. And then along those lines, I was like doing social media, and I decided like back in July of 2022 to make a science channel, and now it's up to like 95,000 followers, which is crazy. People just love hearing me talk about
Wow.
how to get rid of bodies, and it's you know just a normal Tuesday. Like,
That's so cool. That's awesome. So, we were talking backstage, and I just want to start off really quickly with one simple question. We were talking about biogenesis versus abiogenesis. Can you tell us what the difference is?
so I would love to. So abiogenesis is basically the creation of life. So it's like the question about what is the origin of life on Earth. And like we're only talking about Earth here because there are many theories about many other things not regarding Earth, but abiogenesis is the idea that like the origin of life started out with things that weren't alive, of course, because life didn't exist. So it was like inorganic things that created life. In contrast, biogenesis is just the very exact opposite. It's the idea of like certain substances and things like that being created by life. So it's the idea that like only life can create life. So they're very contrasting, but they kind of correlate with the whole idea of evolution and everything like that.
Hey, you want to put a death nail in something for us? So when you're talking about these topics, we talk about abiogenesis, we'll say, "Oh, that was disproven by Louis Pasteur when he did his spontaneous generation experiments." Can you explain the difference between spontaneous generation and abiogenesis for us?
So spontaneous generation is like it says, it's spontaneous generation. There is not time going in between it. So you can't compare like whenever you're doing an in vitro experiment, which is just like in the lab experiment, and you're trying to speed things up, it's going to have its qualms. And so when you're trying to make this like small, like scalable lab in comparison to what we're talking about, which is the entire history of the world right now, like it's gonna have its disparities and you're not gonna have like this exact accurate conclusion. So a lot of the time, like, yes, it can be like technically disproven, but most of the time you would have to have certain aspects of these labs to correlate with the real life version of things.
That's awesome. We do get a lot of, I don't know, people seem to not, well, they don't understand a lot of science.
Yeah.
And then they hear buzzwords that they can kind of correlate with their own worldview, and it kind of gets all muddled and, and then propagated. And that's very frustrating.
It actually, that question reminded me, Bree, of like a conversation or more of an argument that I had with like my ex-boyfriend's dad, like back in like high school. So it was the question of like, if we have not had plastic bags for like the 500 years or so that it says that it takes a plastic bag to degrade in a landfill, like, how do we know it takes that long type thing? And he was just going off on this. And I was like, a respirometer? Like, it was so, a respirometer is just like a way
you don't have to break down the polymer to know how long it takes the polymer to break down.
Yeah, so a respirometer basically tells you how long it takes a polymer to break down. However, it does have its like, because it's in a lab, it's not going to have a very like acutely accurate number most of the time. It's going to have a variation. And so whenever it says, oh, it takes 500 years to break that plastic down, it really means it takes like anywhere between like 300 and 700. Like,
and in the technical papers, you will see that it'll have a plus or minus 25 or something like that. But when someone reports it in pop science, it does that 25 margin doesn't really pan out,
No, because people don't like to read the plus or minus sign.
Right. Or anything sciencey, really. If it gets kind of over their heads a little bit, I think. And so they just want the clickbait or the 5-second synopsis, and that's not how science works.
No, not at all.
That's how you got your 95,000 subscribers, though, is that you make science palpable to someone who hasn't gone to any advanced science courses or hasn't gone. You make it so that like a normal everyday Joe can understand what you're talking about when you talk about especially the forensic stuff, right?
Yeah, like the amount of times, because I have a lot of friends that are not science majors at all, like they are not STEM people. Like one of my best friends is literally an international relations major. Like, so whenever I have to explain these things during my, hyperfixation tangents over weird science shit, it's like, okay, like I have to basically like dumb it down into colloquial like wording in order for her to understand it and be like, oh, I'm on it with you now. Like, so I kind of took that storyteller like framework and used it in the science videos, and now people are asking me to explain like just the basics of chemistry for their chemistry class and stuff.
That's a gift. That's awesome. Not everybody can do that. I wish that more teachers could do that.
I got 2 questions for you to kind of follow up on that that's relevant to what we are talking about here. So, when you're studying biochemistry for forensic purposes, what can you tell about an individual from just a tooth?
From just a
What you?
tooth.
Mm-hmm.
Okay, first of all, just if you have a single tooth, you can usually tell in like your intro to anthropology, like biological anthropology courses that we have to take for the forensic certificates. You can usually tell what type of tooth it is, whether it's premolar or molar, things like that. You can also tell usually what, like how old or how long it was that like how long ago they died. And we even have different parts of skull identification that play into it. So skull identification, you have the 3 like very prominent races whenever it comes to it. Now, here's the thing. In science, it kind of muddles things because in the science world, race is a very, very like large social construct. And so we don't use it a lot of the time. But whenever you're in forensics, you have to because you're actually identifying people. And so in skull identification, things like that, if you have a single tooth, you can actually sometimes identify whether they are of African American descent, of Asian descent, or European descent, and those are the 3 main racial categories that you have whenever you're doing skull identification.
How does that correlate with like haplogroups? Because I know those come into play when you're mapping like genomes and such.
I think you cut out on my end. I'm so sorry.
Oh, you're okay. I was just asking how those groupings correlate with haplogroups.
So that's not something I'm in yet. That's like a more like upper level course that goes into haplogroups and things like that because we have like an entire class over it, but that's for like juniors and seniors. I am a second year.
Well, that's okay. Yeah, we're all learning new stuff all the time. We'd love to have you come back and tell us more once you go to those classes too and tell us about the haplogroups. Because all I know about them is just looking at maps and stuff, just looking at the way they map them, and then they would lay it over like a geographic map to show where the groups came from and things like that. That's interesting though. You can kind of make an inference about. Can you make an inference about like the age of the person as well?
Yes, you can, because whenever you have certain bones and things like that, for one, it's very easy to dictate whether they're a younger person or an older person, because whenever we talk about development, we usually, whenever people are talking about development, they think of it in a very anthropocentric point of view. And so they're like only thinking about the social construct of development and puberty and things like that. But whenever you are doing forensics, you have to think about the ideas of like, development is not only what you see like visually with people, but it's oftentimes the fusing together of certain bones and things like that. So like, for instance, if you're doing skull identification, it's very easy to tell whether it is a child or like a full-grown adult, not only because of size, but you have certain like bone fusings and things like that, such as whenever you're born, for instance, you have 3 plates in your skull instead of just one skull. That helps with the birthing process and things like that. And over time, over the course of your life, especially during puberty, you're going to have those 3 plates fusing together. So whenever it comes down to it, a lot of the time we base that off of like the age of the person, off of like different bone fusings and different developmental stages.
Can you also determine that by the pelvic bones?
So it's easier to determine biological sex with the pelvic bones most of the time, because like women's pelvic bones are typically going to be wider than a man's pelvic bone, like a cisgender man's pelvic bones. And so like it's easier to determine certain things from certain bones. So you're usually, if you have like a pelvic bone, you're going to be able to tell like what age they are through labs and stuff like that. But there are certain things that are just like surface level. You can see it and it's like, okay, this person was a biological man and things like that. So it's
But,
dependent on
but,
what bone you're looking at.
but you can't necessarily tell whether someone from the pelvic bone, whether they say have given birth or not, or if they're older or younger by their pelvic bones. I'm just curious if that's.
So whenever we're talking about like giving birth, that's one of the things where you're looking at like a whole skeleton, right? And you see different like breakdowns of trauma, like trauma to the body. So you'll see that a lot of the time, because usually a lot of times whenever women give birth, sometimes it doesn't go right, sometimes it does go right, like biologically, things like that. And you can actually see little like cracks sometimes with like that will calcify over, and so you can see that calcification. And it's really interesting, like the different like for instance, I broke like 3 bones in my foot like 2 months ago, and like you would be able to see that. like not just because like I have screws in my foot, but also because like the calcification lines are still there.
So you're going to be like, oh, this person, broke their ankle when they were 14. They're 73 when they died. They were, they were buried here for 46 years and all that good stuff. You're going to be able to tell just from the, yeah, that's pretty cool.
They teach you all of it, and then you kind of just have to go into a specialization based off of what part of, studying dead bodies you're most interested in. So.
That is so cool. So I used to, this is so corny, but I used to watch Bones on TV, and that's why I was asking you some of those questions, because I loved that show. And one of the shows, it was like, oh, she's the person was X number of years old and had given birth, and they could tell it because of the pelvic bones and stuff. And I was just curious if that was actually a thing, because you never can trust TV. So I was just curious.
Yeah, shockingly. So it's funny because Bones is very mostly accurate. Like I have watched the entire show, and I'm like, yes. Like I'll literally go into them. Yes,
they get the bug. They get the bugs right, and there's a lot of bugs on Bones, and they get them right all the time. Yeah,
so. They make it cool too.
Bugs are a very, very important part of like forensics. That's like forensics investigations on crime scenes. Bugs are typically one of the most important parts whenever it comes to that, because they tell so much.
I feel a little vindicated now. It's not that cheesy then. If you guys say that it's mostly, obviously it's not going to be completely, but it's mostly accurate, that makes me feel better. I have a connection now. That's cool. So
no, I won't say. Sorry. I was just going to say I won't say that all the bugs they show on the camera are always the ones that they talk about, but yeah, they try though.
They try.
Hey, at least they tried. I mean, there's some that are so way off. You're sitting there looking at the, yelling at the screen, going, "That's not even anywhere near factual. What are you doing?" So
only me though, so it's okay.
So, kind of getting back to our topic, I was curious if we know, and this may seem kind of strange, but how do we know, or what do we know about the first cells of life as reproducing organisms?
So whenever it comes, so we're talking about unicellular life here, right? Because that, like, the origin of life starts with unicellular life. Like, you can't just start with multicellular life. So we have a theory that the first unicellular organism was something called cyanobacteria, which is a very, very simple organism. I'm talking, it has like a strand of DNA and it's just going with it, right? So, but whenever you get into like reproduction, things like that, because most of the time unicellular organisms are asexual, so they can actually reproduce on their own and they don't need another one. Whenever it comes to more like multicellular things, such as like, for instance, a Komodo dragon that I talked about, like fast forward to now, multicellular organisms, super complex. We are talking about multiple different reproduction styles. So for instance, like Komodo dragons actually have something called parthenogenesis, which is basically where they have the option to sexually reproduce and they can do it, but most of the time they reproduce asexually because of the fact that they're actually just too mean to each other to like actually reproduce. So instead of wanting to like reproduce with one another, they're like, I'm just gonna kill you. Like, so it's very
I'll kill you and make more of me.
Exactly. But whenever it comes down to it, like reproduction styles are usually the simpler the organism, the simpler the reproduction style. So that's why you have certain types of bacteria that even now that are just, you know, multiplying. So they're just going through mitosis and just being like, okay, like there's no genetic variation. There's nothing really. It's just quantifiability. How high can we go?
Yeah. Starts off with cloning yourself, and then they clone themselves for a couple 100 million years, and then they decide, hey, if we mix and match our DNA, we survive a little bit better because we don't all die from the same blights and stuff like that, right?
Yeah, and like yes, and like the idea of like going from unicellular to multicellular, because I have a feeling this is going to be a question at some point to me. So there's a theory about that. Mind you, most of science, like people think that science itself is very, you know, this is a definite yes, and this is very fact-based. And the majority of what science is like, you have to come in as a scientist most of the time and be like, I'm taking all of this with a grain of maybe a few grains of salt, like, because most of it is very theory based, but it makes sense. So the theory is going to apply until it's disproven, like very accurately disproven. And we're finding new things out every day, but one of them is if you're mean enough, you eventually, yes. And so if you're mean enough, you may asexually reproduce. No. And now, like, that would be very, very much a ton of evolutionary, like, time. If humans were just like out of nowhere so mean that like either they have the option to like, or they have to have the option to either they're going to go extinct or like they're going to figure it out. I don't know. But yeah. So but whenever we're talking about like unicellular and multicellular in evolution and like the origins of life, we have a theory called the symbiotic theory, which is basically an idea that like, you know, cyanobacteria, for instance, they just got like really close to one another and they just turned into a multicellular organism because that's what makes the most sense, right? And usually the simplest explanation is probably might be right, you know? So that's currently the main theory for that right now.
Do you know Chlamydomus, the algae that we can in the lab, we can switch it between single cell and multicellular.
Yeah.
Yes.
We have a cell culture. So at UT Austin, we have like an algae culture club, and they are obsessed, like absolutely obsessed. The weirder the algae, the like better for them, basically.
It's just, yeah, we've been I follow a lot of like Abogenesis debates and stuff on YouTube. So that's been a talking point that's been brought up quite a bit, is this transition from single cell to multi-cell. Now, if in your opinion, aside from single cell to multi-cell, what's the most significant stage of evolution for to get us to where we are today?
Earlier than the single cell to multi-cell like transition, and that would have to be like the creation or the theory of the creation behind the eukaryote. So for people who just want it in like layman's terms, so eukaryote and prokaryote, two main different types of cells, right? A prokaryote has no nucleus and usually doesn't have like mitochondria either, and a eukaryote does have a nucleus and has mitochondria and has a few different things, but the main two things are, do they have a nucleus and do they have a mitochondria? So one of the most important stages to me, because this literally started everything, is the theory that the like creation of a eukaryote was literally created because a prokaryotic cell had like some bacteria that basically wanted to invade it to kill it, or like to basically have a parasitic relationship with this prokaryotic cell. And so this bacteria went into the prokaryotic cell and then just formed a membrane-bound nucleus and mitochondria. Like it's the funniest thing in the universe, because it's very, if you personify it, you're like, oh my gosh, it wanted to attack this random prokaryotic cell and then decided it's going to be friends with it. Like, but no, so creation of the eukaryote is pretty important and a really funny like theory to me.
And we never would have got to multi-cell or cell division as we see it today if it hadn't cell division in general, right? I mean,
yeah,
it was
so like eukaryotes are basically the start of very complex structures and organisms. Like that's what they are. So like we have our plants and our main animals, and those are both eukaryotes. And then whenever you get into prokaryotes, you're talking about simple bacteria and things like that. So like the beginning of complexity in life started with the eukaryote.
Okay.
Yeah.
So I may be wrong, but haven't they seen in the lab proteins that have actually been able to come together to create a prokaryote?
They create RNA.
RNA. Okay, that's it.
Self-replicating RNA. Yeah.
Okay, thank you. I remember hearing something about it, but I wasn't quite sure.
Yeah, they can replicate it on clay. Sorry, I'll let Melly talk. She's the guest, not me.
No. Please continue. I love this.
Yeah, it's a conversation. There you go. Come on, Bree.
That's just the first. That's the first step for an RNA first world. That's a great discovery, you know. But there's other factors that come into early life. There's a few different hypotheses of how all of that first started, where it started. We were talking backstage a little bit about it. Difference with like thermal vents and wet dry cycles, things like that to help produce and sculpt life into what the myriad that we have today, covering every nook and cranny of this great planet, right?
Yeah.
Yep,
so
absolutely.
Whenever you were talking, you mentioned the RNA like beginnings basically with like abiogenesis, because I just saw a comment right here where it was like, don't confuse a hypothesis with a theory, and I hate that too. Basically, the theory is, because it's a very, very well-known, very popular theory, is that in abiogenesis, the origin of life started with an RNA start, basically. So it was just like, okay, RNA started the entire thing. Before DNA, it was RNA. But there are different hypotheses to kind of combat that theory, because it is the most popular theory that we have right now among scientists. But there are hypotheses that like it was a metabolism start or something like that. So it was pre-RNA start. So that's like the main difference between a theory and a hypothesis, is like the hypothesis is trying to disprove the theory in that case.
Okay.
The leading theory, yeah, and then you got all these hypotheses competing. That's what science is all about, though. That's what you want to do, is try to topple. That's people are like, oh, you get kicked out of the science community if you disagree with their dogma. No, you don't. You get prizes. You get prizes if you're able to turn something over.
Right, no, but they're not. The coolest thing is they're not going to just accept you. Oh, wow, this is the coolest thing. They're going to want to replicate it and test it, and they're going to dissect it, and they're going to chew you up and spit you out, and then they'll find that you're right. And that's a tough thing to go through.
When I tell you the most like shockingly, or maybe not shockingly to most people, but at least shocked me, like everything in science, even the things that we now consider laws like gravity, they have all been some of the most controversial things in their time when they were figured out. And so you have this baseline hypothesis that will turn into a theory, and then you have like so many combating like hypotheses to try to disprove it, because people want to figure out, and their like thirst for knowledge is so great. But on the other hand, it's this idea that like I want to get the credit in that, and I want to like make history basically. And so it's this fight between scientists that I don't think a lot of people realize, because people look at science and they think, oh, this is a fact-based thing that I can trust, and it's like over half the time scientists can't even trust what's happening here.
Everybody wants to be the Newton of their field, right?
Yes.
Absolutely. But the cool thing is that while they may be doing that, they're also doing science. I mean, because you have to be able to falsify, have a falsification, and they're going to try to come up with that and then make that happen. And that's the only way we can learn true things is by trying to do that. So it's very cutthroat, but that's how we actually do science. You don't see that same level with, say, creationism. They may fight among themselves about specific scripture interpretations, but not about like evidence and testing and hypotheses. It's let's look at, you didn't interpret it this way correctly. So that's why I think that science is so important. It may be cutthroat, and it may be that everyone kind of wants to outdo the other, but while they're doing that, they're also being science. They're also creating science. Exactly. And I think that's kind of cool.
Yes, I love how that was phrased. Like,
did you all ever do any studies on slime mold when you were in your courses?
Slime
at
mold?
all?
I'm trying to think back to it. Okay, so in our geoscience class in like high school, we had like a small excerpt over slime mold, and then I've just completely forgotten about it since.
Oh, well, I recommend them. I won't derail the conversation about them, but I think you sounds like you might be interested in learning about them, especially if you like all this, the way that DNA replicates or the way that different organisms reproduce in their own unique ways. You want to see something really weird about reproduction and the way that gametes are shared between organisms. Check out slime molds.
Also,
please.
check out sea slugs. Sea slugs are really like crazy creatures whenever it comes to that stuff.
Oh yeah, slugs in general too. I don't know if you've ever seen the videos of leopard slugs mating, but they make a penis flower. It's quite beautiful, actually.
Yeah, they do.
Oh, how cool. Okay. I got to hear about this. I'm sorry. We're taking a little bit of a derailment. Tell me about this.
Oh yeah, the leopard slugs are hermaphroditic. So, well, they're both male and female, both of them. And the way that pretty much all slugs reproduces, they have love darts, which are tiny little calcified harpoons that they stab into each other. So what they do, the leopard slug specifically, is that the two of them that they'll circle around for a little bit and see if they want to, get it on. They'll like sniff each other's hindquarters and all that, and a little circle for sometimes for hours, and then they'll finally go up to a high spot, and then they'll dangle down from a slime rope, the two of them like intertwined together, stabbing each other in the face with their love darts until it triggers them to extend their translucent white penises and twist them together into a little flower that they could deposit both their gametes into, and mix it all up into like a slurry, and then they pull it back in, and one of them drops down to the ground, the other one goes back up the slime rope, and they go their separate ways. It's quite beautiful.
It's beautiful.
That's so cool.
It's beautiful. However, my favorite part of this, because forensics person always have to bring death into it. Love dart thing. Most of the time, one of the two slugs will die because they will just keep stabbing each other to death in hopes to get it on. So it's it wasn't the basic science stuff as to why I got into it. No, it was the weird stuff.
Yeah.
There's so many jokes, so many things that I could say that I will just not. I love this. That is so cool. All right, so we are going to take a small break because I want to acknowledge all the wonderful people, excuse me, in the chat. So let's see. I'm going to scroll back up here. We have KCA Randy. Good to see you. We have, of course, Kat the Humanist. Look at the trees. The Caribbean Skeptic. Let's see. We have, scrolling down. Sorry, guys. Aaron Coalson. We also have, of course, Skeptic Haven is in there giving some good chats. Azalea Jane. Hey, Azalea Jane. I love your comments. Let's see who else we have. Dan D. Hey, Dan D. Let's see. We also have Phil Calderone is on. We love us some Phil. Let's see. It looks like that's all. So thank you guys so much in the chat. We love having you guys on. And Bree, you want to talk about the shows that are coming up?
For me?
Oh, well, the ones up on the screen Saturday with.
Oh, okay. Yeah. Let me see. Yeah, we got Saturday at 3 p.m. Sorry, I thought it was shilling time.
Sorry.
we got Ben on. Look at that. We got Ben and Heathen Queen scheduled up. What do you want to miss that for? Definitely be here. That's Saturday. Is that both of them on Saturday at 3 p.m. Central Daylight Savings Time? Yes. Oh my gosh, Ben's awesome. Ben's one of my good friends. He's a student, a doctorate or doctor student. He is going medical, so he's going all the way, and he's very active on YouTube and stuff. So if you don't follow Dr. Ben, definitely follow them. But Dr. Ben is going to be live here on Saturday, so don't miss that. We have Heathen Queen, which Jessica's amazing too. And brilliant and kind of snarky. So yeah, always a fun time to hang out with Jessica. So are gender roles necessary? Let's find out. Yeah, show up Saturday. I'm going to be there because, I've filled a few gender roles in my day. So I need to know if I was just wasting my time spinning my wheels. So yeah, we definitely need to check that out. And then what we got? We got Dave Warnock.
I love
also
Dave Warnock.
On Saturday. Yeah. The problem of evil, which is a real problem because, therefore, why evil? Absolutely. And I'm so happy that we still have Dave with us because he had a date where it was all supposed to come to an end. And Dave is still around, still rocking it and still showing up on these little shows and hanging out with all of the skeptical content creators. So I look forward to hearing what Dave has to say about the problem of evil.
Yep, I'm looking forward to that as well. So we'll go to some questions that have been in the chat. So we have one from Phil Calderone. How can new proteins come to be?
How can new proteins come to be? So, okay, whenever we're talking about proteins, we first have to start with like the basic level of proteins. So amino acids that are like tied with polypeptide like bonds, right? Polypeptide bonds. I don't know why I didn't say that right. And I actually talked about this in one of my TikTok videos because there was this like video that I stitched that was like, oh, eating protein is bad for you. And I was like, where are you getting this? But so the creations of a protein is basically building blocks, amino acids that are tied together through polypeptide bonds. And then you have the like 4 stages of just protein folding. But the funny thing is that whenever we're talking about the building of protein, like humans, right, we need protein in order to build protein. We know that from like a diet culture standpoint. But whenever it comes down to it, the reasons we need this is because we have like our bodies basically produce over 500 amino acids that have been identified, and there are still more being identified each and every day. But the main ones that produce protein is the actually just like a list of 20 of them out of those like over 500, and 9 out of those 20 amino acids are the essential amino acids, also known as the ones that you can't create yourself. So whenever we're talking about like protein building, you actually have to consume different amino acids through the protein in your food in order to actually build that protein, going through transcription, translation, and then RNA or amino acid like synthesis, and then that's when the protein folding comes in. But overall, it is just a link of like a total of 20 different amino acids that you can build proteins with, and then it goes through like primary, secondary, tertiary, and like quaternary folding phases, and then that's your protein.
Cool. Okay. Number 2, we have obviously speculative. What do you think the most likely long-standing theory that is commonly accepted today that will be replaced in the next 100 years? Great question, Aaron.
That's a big question. Oh my gosh. Wait, I have to think of like all the theories that we've gone through. So there is a new study coming up, like that came up, where it's talking about the history of, you know, the world and evolution, things like that. And it's this idea that basically, like, there was a time before what, like, when we thought the beginning of the world was. So like we thought it was like the Hadean period, and there's apparently a time before that. So that's basically messing up our entire like worldview of like our like history of the world clock thing. And so now we're kind of, as scientists, like trying to figure out where the missing pieces went. And so I think that's just right now, that's like replacing, that's just like current science that's replacing what we've been teaching like high schoolers for years. So
They're
it's a
replacing
mess.
are they cutting the Hadean period or Hadean era in half? Or are they doing
Apparently it's like before the Hadean period or something. Like, it's a weird thing that I heard about like yesterday, and I didn't really look into it because I was like, this has to be fake news. And then I kind of
What is the
saw more
state of
about it.
matter?
I don't know. Like, I was like, why are we? So basically, the point of that is like there is constantly different things that are making us question the entirety of what we know in science. Like, so whenever it comes to like a specific theory, I could not tell you, but history of the world and how things have happened after like the Big Bang and stuff like that. Like, when we're talking about this history, we're kind of just like mashing all of it together and hoping that we can get like a correct timeline.
That's fascinating. I don't even know what to say.
Do you want to go back to the slugs?
Next question by Dan D. We're the slime molds, the ones they were. Wait.
I think that weir was
using for transportation
auto-corrected.
studies.
I don't know.
Oh,
Were the
we're
slime
the
molds
slime
the
molds.
ones they
Oh,
were
okay,
using
we're
for
the slime molds. That's a good one.
Does anyone know the answer?
I have no idea.
Because I don't know.
Inquiring minds want to know
I need
this,
to like.
though.
I need to up my slime mold like knowledge here. I didn't know this was such an important topic.
I know. I brought it up. I feel like I should hang my head in shame for just. I don't know.
Regret as soon as she like.
Oh my god, that's awesome.
You know what the problem is? It's that we got in Skeptic Haven. There's such a brilliant and smart live chat that all the people out there they're all PhDs and stuff. And yeah. No, there are really some brilliant people that we have in the live chat every time that we do a show over here. So yes. Anyway.
Absolutely. So next question, another one by Phil. Biochemically, how far back do some molecular similarities go from modern species to some very ancient ones? And do you have any examples?
So, okay, molecular, like if we're just talking cell-based, so we all have the same like basic structure of cells. That's what we're all built with. So like I have the same basic structure of a cell as like the fish in my fish tank. Like that's just how it is, because we're all going to have like eukaryotic cells. That's the basic biochemical structure of every single person you meet. But whenever it comes to, because I'm gonna get into like really interesting weird topic here. So part of the evolution like study that you learn as a forensics major or anyone who takes anthropology is like the idea of homologous and analogous structures within people and within different species that kind of link them together. So for instance, like the similarities between people now, we have similarities of like with some, have y'all ever read Your Inner Fish? It's by Neil Shubin. It's like really interesting.
I haven't, but now I want to.
It's so good.
I have not. Yeah.
absolutely love that man. But he actually in 2004 discovered a fish called Tiktaalik, and Tiktaalik has hands.
Yeah.
It has homologous
Oh,
hands to
yeah.
humans. So like a lot of the bone structures in those hands are very, very like we have in common with us. And so it's interesting to see the like evolution similarities whenever you look at different animals and like their skeletal structures.
Didn't
And
didn't Tiktaalik have 7 fingers? I thought it
it
had like
had six fingers.
yeah.
Yeah. And a lot of humans actually have anywhere between 5 or 6 fingers on each hand. It's actually
Polydactyly, yeah.
considered the wild type like allele that people have 6 fingers on each hand, but we just kind of like evolved out of it based off of our mating choices. And so that was like, you know, natural selection going back in and just being like, okay, you're gonna have 5 fingers on each hand now. But so like we
No
have
one
a
warned
lot of
the
different...
AI though. The AI keeps making tons of fingers.
It's like more and more hands. Like, did you see the pictures that AI created of like people shaking hands? Like,
It's like not.
I was like, that's not right. Like, but no,
You're taking a forensic class.
we have a lot of
Melly just
similarities though. It's really cool. And even like there are early like embryonic humans that were studied that were seen to have like gill flaps like in the embryonic stages. And that's not to say like whenever we're currently like modern day humans in embryonic stages that like we become half fish in the middle of nowhere, but that's to say like we have evolved from fish just like we've said to evolve from different primates, we've also evolved from fish. And so it's really cool to see like those linkages historically.
by monophily, we are fish, so you never outgrow your ancestry. So you should. That's why you need to summon your inner fish, and that's what Tiktaalik was all about. The most amazing thing about Tiktaalik is that they predicted where and when, or where they were going to find it, and what layers they were going to find it in, and what features it would have. So fascinating. That is, that working together with the people doing stuff like you're doing, the forensic stuff. So they track down where this sucker is, and then someone like you goes in and studies all the morphology. Of course, you're more towards humans, whether there's people that are studying all these different things. It's amazing. And all the techniques that you use, these same techniques could be used to study a fossil creature that's been buried for millions of years. Amazing.
Yeah, like archaeological paleontologists are crazy, and I absolutely love those people. Actually, that's what Neil Shubin is, I believe. He's like he studies like paleontology and anatomy. Like, it's so cool.
So we have us a super chat. Thank you, Aaron Coalson, $10. Hey, Sunny, glad to see you streaming. Thank you. I took a little bit of a break while we were trying to figure out the new way we were going to do the show. Also, super chats and joining Patreon really help Skeptic Haven produce more great streams like this. Absolutely. Thank you, Aaron. We really appreciate that. And we also have another question. This one also from Phil. He says, clarifying, how do brand new proteins come to be that didn't exist before, evolutionary? What % are good, neutral, bad?
Good, neutral, and bad. I can't really, okay, the good, neutral, bad part, I can't really say just because that's a very vague term in the like whole scope of things. Now, like harmful, you have a lot of proteins that were harmful that were then like, evolutionarily like breeded out kind of thing. But how do brand new proteins come to be that didn't exist before? That is part of the like protein synthesis thing that I was talking about with metabolism. Like brand new proteins that I have not studied because they are not identified yet. Like they'll usually come from those amino acid like block chains. And so it's all about this like building block amino acid thing. And whenever it comes down to it though, it's just like, it's not a different like process itself. It's not a different process. It's just like, you have multiple different amino acids that your body is creating and that is building proteins or not building proteins. And like people have simply just not found them yet. but yeah, that's the idea of it.
Okay, so I have a question because I have also, Bree, been listening to a lot of debates. I don't know what the deal is, but I have just fallen in love with listening to them. And one thing that has come up a lot, oh gosh, it just went right out of my head. Say the last thing you just said you were talking about really quickly, because it brought that up.
Proteins?
Yes,
I was
okay.
talking about the what was
Yes,
yours?
so the gosh. Oh my gosh, it was right there and it went again. It was about proteins. Something that I've heard a lot about. Okay, let's move on and it will hit me all of a sudden. I can't believe I can't remember what it is. It was right there and then as I was speaking, it went away. That's terrible.
Was it
I
about
hate that.
telomeres?
And what was that?
Was it about telomeres?
No.
Okay.
No, it was it will come to me in just a second. I hate it when that happens, when my brain does this to me. It's like it's trying to go against me and I don't like that. It's not fair. I want to say really quickly, theoretically is in and apostasy. Hey, Stacy. Love it. Stacy says, Sunny, I hate when that happens. It will come back. Please tell me it will because I hate that when it happens.
Well,
It
if
was
you
right
gotta
there.
stop trying to think of it, the first thing you gotta do is clear your mind.
It'll
and stop
come
trying
back
to...
as soon as you like go into another topic. Okay,
That's
wait.
right. That's exactly right. It will come back to me. I'm sure it will. So go ahead.
When it comes to famous cases of thawed out remains like Otzi, the Iceman comes to mind. What can we, I guess, what can we glean from that, or what would we expect to be lost from a body that age? Or would we be able to get DNA to map what he looks like? Will we be able to clone Otzi someday?
So I want so badly, because here's the thing, like this is like a topic in all of my classes. We've been talking about it and we're like, we want him to like be cloned. Like, let's do it. Yes. Whenever it comes down to it, because like freezing an organism is so much different than just like its natural decomposition process, because you're adding in this frozen kind of like time freezing like thing whenever it comes down to like the life of the cells and things like that. And you have different species that'll react differently to that. So like for instance, like OC is like a mammal and like we have like different frogs that can randomly just like, you know, be frozen, not die, and then like come back like nothing happened whenever it thaws out. And so but I can say that the DNA is basically like frozen. Like those cells have frozen in time. I'm not saying that they didn't die in the process and that the main functioning ones did not die in the process, but I am saying that DNA is still there. So it could be possible, but it would be very, very like difficult to clone Otzi.
I'd just be interested to know what kind of like phenotypes and phenotypic stuff that you can't see. Did we lose you, Melly? Oh, there you are. Okay, you froze for a second.
Phenotypic stuff that you can't see, and I was like, and it cut out, and I was like, great. Like,
yeah, that was taken in the decomposition process. Some of the phenotypic features of his face and his body and stuff like that. I just want to know what he looked like, I suppose.
Same here, honestly. Like the amount of times that we're like I'll look at like a cadaver or something, and like my classmates will be like, God, that decomposition did you dirty. Like it's so bad.
So yeah, you have there's like corpses in front of you all the time. Is that?
It's not all the time. It's very rare actually, because like, you know, most of the time in order to like be with a corpse, you have to like have a certain amount of expertise and a certain amount of experience along that. And so like we'll see them, and at this point I'm just like very emotionally numb to it because I'm like, okay, they're not alive, which I would be freaking out if I was in the pre-med space, because if people were alive and I was just like messing with their bodies, that would scare the crap out of me. But because I'm like, oh my God, I could hurt you at any time. But whenever you're looking at like a dead cadaver that's just like there, you're just kind of like zoned out. It's the same as like identifying random bones. Like you take the personification and like the human out of it, and you're just like left with this like mass, like this pile of matter.
I just remember my question.
Oh, go ahead. Go ahead with your question.
It came back to me. It was like a lightning bolt, just kind of. Whoosh. So in in the debates, I hear a lot about the codes in DNA. That it's that it's coding for things. There's this great code that only a brain could or a mind could put into that, and it drives me up the wall. So can you tell us the difference between what we would think about with computer code? Not that you would know much about computer code, but genetic code. What in the world are they talking about?
Okay. My favorite, love genetics. This is like the best topic ever. So whenever you have DNA, it's a double helix, right? So we know what a double helix looks like. You see it in like random science shops and you're like, haha, that looks cool. So that DNA, those like double helixes have what we like to call nitrogenous bases. And those nitrogenous bases on this DNA is going to be either adenine, thymine, cyanine, or guanine. And those are like the 4 nitrogenous bases. I'm not going to go into the structures because I'll nerd tangent. Anyway, so you have the 4 bases in DNA. And those bases are basically connected through this like, I lost it for a minute there. I'm getting what you got, Sunny.
It's contagious. I'm so sorry.
I was and then I just lost it. So, okay, basically these nitrogenous bases are connected with like a molecular backbone is what I'm going to call it, because I forgot the other word. So molecular backbone is what connects these, and that's what makes the double helix shape. And these nitrogenous bases connect in pairs. So you have thymine that's connected to adenine, and then guanine that's connected to cytosine, cytosine. And so whenever these are connected in the transcription process, you have, you're creating RNA from that DNA. And RNA is the same-ish, except for instead of thymine, it has uranine or uracil. Starts with a U, uracil, I think. So we just know them as A, G, C, T, and U. Like that's what we do. And so I'm like, what are the names of these things? But instead of T, you have U. So U and A connect whenever you're doing like RNA. But RNA is not a double helix, it's a single helix. So it only has one strand. So you're taking that RNA, you're like copying the like complementary bases from the DNA, and then through like, lagging strands, leading strands, things like that, we go into this like DNA replication phase. But right now we're talking about transcription. So you have this whole thing happen to where you have a single strand of RNA that has these nitrogenous bases that's, does not have T but has U in it. Those nitrogenous bases are what make up the code. So you have 3 nitrogenous bases at a time that go through a ribosome. And a ribosome is just like this little dot thing that this RNA goes through, and it goes through and it has like whenever it goes in with the nitrogenous bases, it comes out with an amino acid that goes with that code. So you have like AUG. Yeah. So you have the starter of like, for instance, every amino acid chain will usually have the same starter, which is AUG. I believe that's it. Yeah. AUG, which will then code for methionine, which is one of those 9 essential amino acids that like you can't produce yourself. You have to, get it from another protein source. But you are creating these, like you have these 3 nitrogenous base codes that will then go through this ribosome and come out with a different amino acid each time. And some of the amino acids have multiple different codes. Some of them have like just one code that correlates with them. And then you have a code for like the stop codon, basically. By the way, those 3 nitrogenous bases are called codons, which is super fun and, creative. But
makes it easy to remember though.
Yeah.
That's what I like about codons.
And so that's like code. It's not like this computer code where it's just a ton of zeros and ones. It's like a very different type of code that doesn't even involve numbers over half the time.
Thank you so much. I was. It always drives me crazy when I hear that. I always think we kind of give it the name or describe it as code because we're trying to correlate it to something that we can understand.
Yeah.
And so we say code, but it's really the connections and like they're attracted to each other. So if you have those that what was it, UAG?
So if you have like AUG,
AUG, that's it.
It goes into a ribosome as an input, and then what comes out is an amino acid that, like methionine, which is the amino acid that was coded from AUG.
Mm-hmm. And so I just think that's so fascinating. I absolutely love science.
I'll be right back.
Okay. We
Sorry.
are getting close to being over time, but if you don't mind, there are just 2 more questions in the chat. Is that okay?
I'll see if I can answer them.
All right. So the first one is, I just love this. Thank you, Aaron. Thoughts on bringing back the woolly mammoth?
Oh, okay. So this isn't very sciencey of me. It's just very nerdy of me. I want to bring back this woolly mammoth so badly, but also I have to think about the fact that climate change is like an impending doom about us all, and a woolly mammoth would just not survive in these conditions. So like if we have polar bears barely surviving in the Arctic because the Arctic is heating up 5 times faster than the rest of the like globe right now, like woolly mammoth would probably not survive as soon as it got like cloned, basically. But I really do want them to come back because I absolutely love them.
So that might be a campaign to stop global climate change, man-induced climate change, because we want to bring back the woolly mammoth. Come on, guys. Let's make this a better world, right?
Absolutely. Oh my gosh, that's the next Instagram post.
There you go. We have the slogan. And the last question is from the Caribbean Skeptic Secularist. And he asks, how is the evolution of Archaeobacteria? I cannot read today. Archaeobacteria different.
Archaeobacteria is different from Eubacteria. Which came first, the archaea or the eau? I actually do not know. I have not studied like specific bacterias. It's more like you just have a baseline of like surface level knowledge that you're given about these things. And the reason that we even use cyanobacteria over half the time for the theory of how like, you know, like the evolution of life is because that's the one that's very like that has the most evidence towards it as to like that's like one of the oldest unicellular species. And so, but whenever it comes to different types of bacteria, I would go to a microbiologist for that.
Well, we also know, like you said, hey, welcome back, Bree. So while you were gone, we decided that we have a new campaign slogan for climate change, to reverse climate change, and that's bring back the woolly mammoth.
Yeah.
Make the world a better place.
You know, but people talk about, I do like Pleistocene rewilding in theory. I just think there's a lot of other ecological projects that we could throw money into before we start doing that. Restoring some of the more beneficial to the climate, some of those ecosystems. But yeah,
we have to make anything survive anymore. We're not going to survive if we don't do something.
Right. Yeah, I'm with you.
Kat says, I think we should breed tardigrades. They're 100 times bigger.
Oh gosh, that would be terrifying. Do you understand how invincible those things are? Like, they rule the world and we couldn't stop them.
So, do you know anything about the beetles the beetle larvae that are breaking down plastic now?
Yes. So we have this these studies and like these research projects and like the University of Texas Austin, and that's one of the things that people are really talking about. But that, along with the mycology freaks who really, I'm a mycology freak, so I can call them that, felt that. But like whenever it comes to the degradation of plastic topic, like a lot of people go towards the like 50 different species of mushroom and fungi that can degrade certain types of plastics, but the beetle larvae is like this newest like craze that I'm still obsessed with.
It's styrofoam that they're focused on too, and that's a really hard polymer to.
Thank God, because the amount of like mycological findings, and we have not found a single species. It's always like the weird plastics that like no one wants to degrade, but like whenever it comes to like polystyrene and certain like polyethylene, like
polypropylene,
specified polyethylenes, like you really just want to, you know, break those down.
Phil just popped in to say hello.
Hello, Phil. So, I did not know you were a mycology nerd. This conversation could have went way different if I had known that.
I was going like, before forensics even came to be in my brain. I was like, I'm gonna be a mycologist. We're doing that, and then like.
Well, see, that just means that you need to come back on again.
I do.
That's what that means. So we have gone over, and I do appreciate you allowing us to do that because I do respect your time, but we really want to make sure we get to everybody's questions. And it was so fun. It seemed like we just started. I mean, I can't believe it's over already.
I could continue this conversation for like 2 more hours. Crazy.
I know. And I could listen to you for 2 more hours. So do you have any final comments or anything that you want to say to kind of wrap it all up?
So follow me on my platforms. I post about weird science stuff all the time, and you will not bored ever. I just talked about like a hate circuit that we have in our brains that like, you know, it's super fun. So follow me on all of my platforms, and also what I tell all of my, you know, forensics freaks out there on my channel. Disclaimer, this is solely for educational purposes. Please do not use any of these methods to do anything illegal on my behalf. So have fun with the information as you will.
Dang, I was so looking forward to doing something illegal on your behalf, but but do check her out. She's she has some really great things. She's on TikTok, and she's doing some really great work.
She also has a great smile, I just want to say. So
she does. You are so right. Absolutely. And so Bree, what are you having coming up?
So I live from the Hive Saturday mornings, 9:30 a.m. Central Standard, or Central, I guess it'd be Central Daylight Savings now, but Central Standard soon enough. And we'll do this week. What do we got? Since it's St. Patrick's Day Friday, I'm going to debunk some fairy videos for Cryptic Corner. We are going to in Creation Watch. I actually am going to be. I'm for a change. I'm targeting the pagans, so that'll be something different. We're going to be talking about with some fairy forests and stuff that people claim are, and there's mycology is going to come into play because a lot of the stuff that people talk about with forests, fae and fairies are the results of mycology, and maybe the mycelial network was the fairy kingdom the whole time. But so come for that on Saturday. Thank you.
Awesome. Well, I want to first thank all of our viewers. Without you, we wouldn't be able to do this. I want to thank all of our wonderful people in the chat. You were great. We absolutely adore you. Be sure to like, subscribe, share, hit the bell. And if you really want to help us out, join our Patreon. You get special perks. I want to thank our current patrons. I always do that. Deneen Murphy, Dan D, Tara S, Michael Wiseman, Kathy Cotton, Cindy Plaza, Aaron Coalson, Phil Calderone, Deborah Grace, and me, Sunny Shell. I want to thank our producers, Wes, Phil, and Kat, and the booking team, Wes, Raymond, Ellie, James, and Correa. Remember, we have other shows on here, so check them out. We have Secular Soapbox, Women Atheist Unload, Global Atheist, and The Bible Says What. So until next time, remember to always think critically. Good night, everybody.